unraveling the secrets of ancient mammoth microbes. Imagine unlocking the mysteries of diseases that plagued creatures from 1 million years ago. A groundbreaking study has done just that, shedding light on the microbial world of woolly and steppe mammoths. The research team, led by Benjamin Guinet, a postdoc at the Center for Paleogenetics in Stockholm, has discovered microbial DNA preserved within these ancient remains dating back over 1 million years. But here's where it gets controversial. Some of these bacteria were not just post-mortem invaders, but companions to the mammoths during their lifetimes. By analyzing DNA from a diverse range of specimens, the team sequenced 440 of them for the first time. From a steppe mammoth that roamed the earth approximately 1.1 million years ago, they even managed to piece together parts of bacterial genomes, offering a glimpse into the infections or inhabitants of these prehistoric giants. Ancient microbes, modern insights. Guinet and his colleagues have pushed the boundaries of microbial DNA study, extending it beyond 1 million years. This opens up exciting possibilities to explore how microbes evolved alongside their hosts over time. The project involved an international collaboration with experts working across multiple collections and laboratories. Strict laboratory protocols, computational filters, and DNA damage checks were employed to distinguish life-stage microbes from post-mortem contaminants. This meticulous approach allowed the researchers to focus on the microbes that were likely part of the mammoth's internal ecosystem. Unveiling the mammoth's microbial community. The scientists consistently found 6 types of bacteria across different mammoth remains, including close relatives of Actinobacillus, Pasteurella, Streptococcus, and Erysipelothrix. This pattern suggests a long-standing relationship between these microbes and mammoths, enduring environmental changes and population shifts. The team distinguished between host-associated microbes, those living within or on the host during its lifetime, and environmental bacteria that entered after burial. Using metagenomics, a method that reads all DNA in a sample simultaneously, they identified ancient damage patterns to authenticate the oldest material. From the 1.1 million-year-old steppe mammoth, the researchers reconstructed partial genomes of Erysipelothrix, likely from an infection during the mammoth's life. This is the oldest authenticated host-associated microbial DNA ever reported, expanding our understanding of ancient host-microbe interactions. The significance of ancient microbes. This discovery builds upon previous work that recovered genome-scale data from mammoth teeth over 1 million years old. It demonstrates that DNA can survive under the right conditions, even in extreme ages. Ancient DNA is fragile, breaking into tiny pieces, and chemical changes accumulate over time. By tracking these signatures, scientists can distinguish ancient sequences from modern contamination, ensuring the accuracy of their findings. As microbes evolve rapidly, obtaining reliable DNA data across more than 1 million years was like following a trail that kept rewriting itself. Explained study senior author Tom van der Valk. Our findings show that ancient remains can provide biological insights beyond the host genome, offering perspectives on how microbes influenced adaptation, disease, and extinction in Pleistocene ecosystems. The Pleistocene period, spanning from about 2.6 million years ago to 11,700 years ago, was characterized by repeated ice ages. Microbes in mammoth health. One bacterium identified in the dataset is closely related to a Pasteurella species implicated in lethal outbreaks among African elephants. This connection is supported by a 2023 paper that identified a Pasteurella relative as the likely cause of fatal septicemia in elephants in Zimbabwe. The new study reports putative virulence factors, gene features associated with disease-causing ability in some clades. The authors exercise caution regarding cause and effect due to the incompleteness of ancient DNA and the mixed traces left by infections. Erysipelothrix, another bacterium found in the dataset, is known to cause illness in Arctic wildlife, including muskoxen during unusual mortality events. This pattern has been documented in a Canadian veterinary journal, which found Erysipelothrix ruseopathii associated with widespread deaths in the Canadian Arctic. Microbes and mammoth survival. The persistence of certain microbes with mammoths for hundreds of thousands of years hints at stable relationships shaped by diet, climate, and social behavior. The presence of these lineages across distant regions points to deep coevolution. The last woolly mammoths survived on Wrangel Island until approximately 4,000 years ago, long after mainland populations had vanished. Genetic work on this relict group suggests that their late survival was linked to small population size and the accumulation of harmful mutations. This work opens a new chapter in understanding the biology of extinct species. Not only can we study the genomes of mammoths themselves, but we can now explore the microbial communities that lived within them, said Love Delane, professor of evolutionary genomics at the CPG. The future of this research, reconstructing ancient microbiomes, can help establish baselines for living elephant microbiomes. This is crucial as health, diet, and stress are all reflected in the microbiome, the community of microbes living within and on an organism. Ancient pathogen signals can also clarify the role of outbreaks in population declines. Identifying which bacteria carried disease-associated genes in the past could inform surveillance and treatment strategies for modern elephants. Additionally, applying these tools to other Ice Age species can test whether long-lived microbial partnerships were the norm rather than the exception. Careful sampling, clean laboratories, and independent checks will ensure the validity of future claims. The study is published in the prestigious journal Cell. Stay tuned for more fascinating insights into the ancient world and its connections to our modern understanding of biology and disease.